gastric pits, which tend to be very deep pits in the pyloric glands, deeper pits than for the cardiac or proper gastric glands. These are the gastric glands here that are invaginated from the base of the pit here at the neck down to near the lamina muscularis mucosa. So they are contained within the tunica mucosa, so they are mucosal glands. They're simple coiled tubular glands, and they are mucus secreting with some inactive parietal cells here. Okay, so that's just a quick review. Now, moving over, let's zoom out. And about right here, you'll see that we had a fairly thin tunica submucosa on the gastric side of things. It looks a little thicker here. This is a mucosal fold in the stomach. That's called a rugae, plural. Okay, so we have a fairly thin submucosa on the gastric side of things, but wow, look at this. This submucosa is greatly expanded on the small intestine side of things. Okay, and why is that? Well, it's because it has these submucosal glands. So remember, the digestive system likes to have a shot of mucus when the food bolus enters the esophagus. So we have submucosal glands there, and they're mucus secreting up in that cranial portion of the esophagus. Then the body likes a jet of mucus when the food enters the glandular stomach at the cardia. So those cardiac glands are mucus secreting. And then it likes mucus to help that chyme be lubricated when it moves into the small intestine. So the pyloric glands are mucus secreting. And then we have submucosal glands, which are mucus secreting at the beginning of the small intestine. And the chyme, when it leaves the stomach, is very acidic and pretty thick. And so we need to lubricate it. And then we need to change that pH pretty rapidly. The way we do that is we secrete a watery fluid that is loaded with bicarbonate. Well, who does that? That's the intestinal glands, the small intestinal glands, okay? So if you zoom out, you'll see on the surface of the small intestine, you'll see these evaginations with an E extending out into the lumen. And then you'll see invaginations extending at the base of the villus. So these evaginations are villi. Okay, it's a way to increase surface area. At their base is where the intestinal glands empty out. So these are the intestinal glands. They're just like the gastric glands. They're invaginated down into the lamina propria. They do not extend past the lamina muscularis mucosa. Okay, so intestinal glands are mucosal glands. And these glands are secretory. They secrete a watery fluid that has some mucus in it from the goblet cells, and it has a lot of bicarbonate in it. And that bicarbonate is going to raise the pH of the acidic chyme, because the pancreatic enzymes that are released into the duodenum, remember, either at the accessory pancreatic duct or the pancreatic duct, those enzymes need a more neutral pH to become active. Now, the submucosal glands here, they do penetrate through the lamina muscularis mucosa. So you can see right here, they're passing through or penetrating that lamina muscularis mucosa. And they will either duct themselves up, empty up into the base of the villi, just like the intestinal glands do, or sometimes they'll empty into the fundus of the intestinal glands. Remember, fundus means furthest from the opening or the base. So this is the fundus of the intestinal glands. And sometimes those submucosal glands will empty right into the fundus of the intestinal gland. Here we can see the lumen of the intestinal gland. And these are called enterocytes. So that the simple columnar epithelium that lines the intestinal glands and lines the surface of the villi, those are called enterocytes. And of course, there are goblet cells. There are also enteroendocrine cells. Here's one right here, actually. Usually you have to have special stains to see them, but there's a little bit of one right here. Sometimes we can see them without special stains. So these are kind of burgundy kind of granules. So these would be enteroendocrine cells. So enterocytes are the simple columnar cells. Goblet cells are the mucus secreting unicellular glands. We have enteroendocrine cells, which we rarely get to see. And these are really cool. This is a section of gastroduodenal junction from a cat. And cats have these globule leukocytes. So these are actually white blood cells of the cat. And we don't entirely understand their function. We think they are mast cell lineage. We think they are increased during intestinal parasitism. And there are papers documenting tumors that arise from these globule leukocytes, which I don't think are in the notes. I don't think you have to ID these globule leukocytes, but I thought they were way cool, and I would point them out to you in this feline section. Okay, let's see. So what is the summary of what the small intestine does? Well, we need mucus, so we need to lubricate the chyme as it comes in. The source of mucus is either these goblet cells in the epithelium of the villi and of the intestinal glands, or the mucus can come from the submucosal glands. We also need to start digestion. And how do we do that? We secrete intestinal juice, and that is what the small intestinal glands do. They secrete intestinal juice, and that watery bicarbonate-rich fluid will neutralize the pH so that the pancreatic enzymes can become active. There are also membrane-bound enzymes. So the pancreas releases free enzymes, but there are membrane-bound enzymes on these enterocytes, and they're bound up here at the microvillus surface. So they also aid with digestion. Okay, so this is the microvillus surface right here. And this is called the striated border in the small intestine. Now, of course, there is a lot of absorption by these enterocytes. So the intestinal glands do a lot of secretion, but the enterocytes lining the villi do a lot of absorption. And they will absorb fats and package them into chylomicrons. They'll release those chylomicrons into the interstitium here, and those will be uptaken into the lymphatic capillaries. These lymphatic capillaries are usually located in the middle or centrally within the villi, within the lamina propria of the villi. So they'll be called central lacteals, and that is the centrally positioned lymphatic capillary where fats are absorbed. You won't see it in every villus. Sometimes you see them, sometimes you don't. It depends on the section and whether they are distended or not with the fatty chylomicrons that they have absorbed. Look here, there's two more endocrine cells. Here's one and here's one. Also, we have blood capillaries. So there are two types of capillary beds within the lamina propria of the villus. There are blood capillaries for absorbing proteins and carbohydrates, and then the central lacteal for absorbing lipids. Okay, so that's for the gastroduodenal junction. Let's look at, nope, that's not what I want. Oh, I put it over here. Sorry. There we go. Let's look at another slide. This is 94. We have lots of examples of small intestine, so it's just for you to practice. Here's a little bit of MALT, mucosa-associated lymphoid tissue, more specifically GALT. So these look like secondary nodules with germinal centers, mantles. That's the B cell population. Here's the T cell population, the diffuse portion of this lymphoid tissue. Here are the villi that you can see in various planes of section. You know, it's like a shade carpet, and they'll have all different kinds of profiles. Some of them won't look like they're attached down to the rest of the small intestine, but they are. That's just sectioning artifact. This was bent so that you didn't get the entire villus when the knife blade went through. These are smooth muscle slips from the lamina muscularis mucosa. So here's the lamina muscularis mucosa. It's very well developed in the small intestine and even has two layers. Here's an inner circular layer and an outer longitudinal layer. Smooth muscle. Muscle slips come up from that lamina muscularis mucosa and extend into that villus. It is thought that when these smooth muscle slips contract, you see how it would shorten the villus, and it's thought that that would help pump out nutrients from the engorged lymphatic and blood capillaries after digestion. These smooth muscle slips will contract and shorten the villi and help pump out those fluids, nutrient-rich fluids, down into the larger underlying blood vessels and in the submucosa, and leading on out into the general circulation, really headed to the portal vein. The blood capillaries eventually will empty to the portal vein to go to the liver for further processing, and then the lymphatic capillaries will ultimately drain to the cisterna chyli, and then that thoracic duct will drain the cisterna chyli that you saw down in gross anatomy. There's a fun something in here. I think it's this slide had a cool little, maybe it was another slide. One of these had a little parasite that I was going to show you. I don't think it's this one, but we'll find it. Okay, so this was slide 94, an example of small intestine. Slide 93, yeah, this is the one that had a fun little parasite. See that right there? That he's pretending. He's saying, I'm a villus, don't pay any attention to me. But that's not a villus. That's a parasite. That's an intestinal parasite. Isn't that way cool? You learn about those in parasitology next year. Okay, so more villi. These are sectional artifacts where you get kind of a transverse or cross section of the villi. I like those because that helps you find the central lacteal if the central lacteal is going to be visible. Sometimes they aren't. You have to really move around quickly and just kind of look for a clear space that might be a central lacteal. And of course, when you're looking for one, that's when it's hardest to find it. Here's a central lacteal here. It's got a little bit of proteinaceous material. It's kind of a blue-gray color. And then there are blood capillaries here. There's a little bit of a central lacteal there. Central lacteals and blood capillaries, of course, will be lined by an endothelium. Let's see, we looked at 94. This is 93. These are just, you know, examples of a small intestine, multiple examples. 57, so repetition is good. That's why we've got so many slides. We also have so many slides because of species differences, and also there's duodenum, there's ileum, and jejunum. So there are a lot of different places to get samples of the small intestine. Now, if you see some mucosal glands like this right here, that's one way that you know where you are in the small intestine, because these submucosal glands are also called glands of Brunner. They're located in the duodenum. So when I see these submucosal glands, even if I don't have a gastroduodenal junction to let me know where I am, I know this is a section of duodenum. This happens to be equine duodenum. These are really nice submucosal glands where you can see the mucin granules in the cytoplasm, so they're mucus secreting. And we have this slide to show you. This is an equine, and we wanted you to see these Paneth cells, cells of Paneth. They're also called acidophilic granular cells. Dr. Paneth identified them. They secrete peptidases and lysozymes. We find them in horses and ruminants. Horses and ruminants have these acidophilic granular cells, and they'll be down here in the fundus of the intestinal glands, and they release their enzyme, proenzyme packages into the lumen. Okay. Now, another name for intestinal glands is the crypts of Lieberkühn, crypts of Lieberkühn. So Dr. Lieberkühn found these small intestinal glands, identified them, and their invaginations at the base of the villi, so he called them crypts. So sometimes in literature, you'll read about intestinal crypts or the crypts of Lieberkühn. That's just another name for intestinal glands. Something that's way cool about this horse slide is not only are the acidophilic granular cells or Paneth cells really spectacular, but also remember when we looked at blood smears for the horse and they had those eosinophils with the large mulberry granules. Well, here they are. Now, these are not the same thing as the cat's globule leukocytes. These are just horse eosinophils. And it's very normal to have quite a lot of these eosinophils migrating around in the small intestine. I took this slide to a pathologist and looked at it with him just to find out if he thought maybe this horse had, you know, a high intestinal parasite load, because there were so many eosinophils and they seem to be associated with intestinal parasitism. But he said, no, that was just a horse thing, that they really like to circulate their eosinophils in the walls of the intestines. So that was interesting. So that's what this slide is for, to demonstrate. And let's see, I think there was one more small intestine slide here. This one helps demonstrate the multiple ways that we increase the surface area in the small intestine. One of the ways are these circular folds called plicae circulares, or circular folds. So this increases surface area. Now these are not permanent folds. They come and go depending on the distension of the small intestine. So these are kind of analogous to the rugae. When the stomach is empty, it wrinkles up, the mucosa wrinkles up on that loose underlying submucosa and forms those folds, or rugae. These mucosa folds are called plicae circulare. They can form because the mucosa can slide easily on this loose pliable submucosa. And again, they'll go away when the lumen is distended. Two other ways that we increase surface area, of course, are the villi and then the microvilli. These are intestinal glands here. They're really long and deep on these plicae circulare. So intestinal glands can vary in height. Let's zoom in if we can see the striated border on this one. It's not a very good slide. Well, here we go. Here's a great striated border right here. So be sure you get it in your mind that these are microvilli, or the striated border. And then the villi are the larger projections, okay? Both of those are a way to increase surface area. I don't know if Dr. K talked to you about well, I know he did. I know he talked about the junctional complexes way back at the beginning of the class, but I don't know if you talked about terminal bars and terminal webs, but these little dots here are terminal bars. They're just junctional complexes like zonula occludens and zonula adherens and macula densa. And then the long line there, that's just cytoskeleton piled up right there, up under those microvilli, okay? All right, so moving on to large intestine. The large intestine, for one, has a lot more GALT. So here we see a lot more GALT. And then take a look at this. There's no villi here, okay? So let me move to a small intestine slide again. See the villi? See all the villi projecting into the lumen? See there's spaces around these finger-like projections? Okay, those are villi with small intestinal glands at their base. But this, okay, there's no villi here. Now there's a little gap, a little divot here, but that's because that's where the villi don't form over the craters of the domes of the GALT, because we want to get antigens down there for the M cells, the microfold cells, to have access to. So if we zoom in here, these are not villi, okay? So this is a smooth luminal surface, and there are invaginations down into the lamina propria. So you ought to be thinking, oh, well, I've got invaginations down of the epithelium down into the lamina propria, down but stopping at the lamina muscularis. So there's only one thing that can be, those are mucosal glands. These are simple, straight, tubular glands. And these are the large intestinal glands. And you can see that they are super duper mucus secreting glands. And that makes sense because when the ingesta gets to this point in the large intestine, what are we doing? We're absorbing. We're not really secreting at this point. We're absorbing extra fluids. We're lubricating the ingesta, preparing it to be passed as a fecal mass. And we're also absorbing ions, minerals, and vitamins at this point. So these enterocytes that are actually hard to identify, the simple columnar cells, the enterocytes, are kind of crowded out, hard to see for all the massive amount or volume of goblet cells here where we're secreting that mucus. We don't have submucosal glands, so we got to secrete the mucus some other way, and that is with a lot of goblet cells, okay, and no villi. Now there's an H&E to show you the large intestine, and there's a lot of sectioning artifact here. And the secret to large intestinal glands, when you've got the whole slide, is drive around and find a place where there's no artifact, no sectioning artifact, where you can see where these are truly just simple tubular glands. Now, sometimes the very beginning of that intestinal gland will kind of artifactually pull apart like this, but that's not a gastric pit. Gastric pits lead into, if the glands were this short and if the glands were mucus secreting, which would only be cardiac glands and pyloric glands, those glands would be coiled. They would never be simple tubules like this. So that's a major, major clue that that little sectioning artifact there is actually not a pit. It's just the expanded lumen of this intestinal gland, large intestinal. Woo, bless you. All right, so now that ends the digestive tubule. Let's just get an introduction to the extratubular glands, the pancreas, just some basic concepts here, and then we'll expand upon those basic concepts for the next lab. I just don't want you to have too much new stuff on that last lab before the midterm. So I want to just go ahead and get an early introduction to the extratubular glands. This is the pancreas. You've probably seen it down in the anatomy lab. It's got lobes. Remember, there was a right lobe, a left lobe, and a body. The lobes have lobules. So the stroma of the gland is the capsule, and then the septa. And the septa subdivide the lobes into lobules. So here are little lobules. And wherever you see this kind of line here and a line here, I'm going to outline one little lobule right here. Okay, that's the parenchyma, the stroma. So the stroma is the capsule and the septa. Then the parenchyma is the functional portion of the gland, and that includes the adenomeres, which are the secretory units, and the duct system. So adenomeres and ducts. So at low power mag, when I drive around, what are these circular structures and transverse sections that have lumens? Well, let's see. This is a muscular artery. This is its partner, the vein. Okay, here are some unmyelinated nerve fascicles hanging out with that artery and vein. If we looked around, we'd find some lymphatics. Now, if we come down here, this is not a blood vessel. If you look at the epithelium, you see that that's a simple columnar epithelium. So definitely not a blood vessel. So this is a duct. This is an exocrine gland, and it has to duct its secretory product to the lumen of the small intestine, to the duodenum. Okay, so this is inter, I-N-T-E-R, lobular duct, because it's between the lobules. If here's an interlobular duct, a smaller one. Okay, so if there are I-N-T-E-R, interlobular ducts, then there are intralobular ducts within the lobules. Okay, so let's zoom into a lobule. Here's a lobule right here. And we're going to use this lobule to take a look at some things. Here are blood vessels, smaller blood vessels within the lobule. So this is the artery, this is the vein. They're filled with blood, and the blood is kind of homogeneous staining, and you can't see the individual red blood cells, but that's okay. That's an artery and a vein. Here's a smaller artery right here. Okay, here's a smaller artery and vein right here. These are the islets, the pancreatic islets. This is the endocrine portion of the gland. We won't talk about this until the endocrine section, but we will point these out, pancreatic islets. Well, then what is this? Well, this is a duct. This is an intralobular duct, a duct within the lobule, intralobular duct, and that's in longitudinal profile. If we come over here, there were some in cross-sectional profile that I wanted you to see. So naturally, it's not jumping out. Let's see. There's an intralobular duct here. Oh, for goodness sake. There were some beauties in here. All right, they're not jumping out at me right now, but I'll find them. So the intralobular ducts start with a smaller duct called the intercalated duct. Okay, intercalated ducts, here we go. Intercalated ducts are real tiny. So this is an intercalated duct. That's the first section of duct outside of the adenomere, and then they become intralobular ducts. intercalated or smaller, about the diameter of the individual epithelial cells. This is an intercalated duct right here. Okay, so these are the epithelial cells that make up the adenomeres. The adenomeres are the secretory units. This is a compound tubuloacinar gland. Tubuloacinar refers to the shape of the adenomeres, little short stubby tubes with a round ending on it. Acinus refers to something that's round. So tubuloacinar and that rounded portion is right here. So we call these epithelial cells that line that adenomere, we call them acinar cells. Acinar cells. So this is an individual acinar cell right here. Extremely basophilic cytoplasm, very euchromatic nucleus. Here's lots of proenzymes called zymogen granules, because that's a generic term for stored digestive enzymes, proenzymes. These are protein-making machines, these acinar cells. They've got a lot of RNA in their cytoplasm. They need that to make all of these proteins. The beginning of the duct system is called the central acinar cell because it's in the middle of the acinus, so centroacinar cell. It's got this incredibly euchromatic nucleus. I like to think of it as like little twinkling stars here in the middle of the adenomere. So central acinar cells start, it's the beginning of the duct system that's kind of invaginated down into that adenomere. So the duct system kind of starts inside the adenomere. Then just as soon as you get out of the adenomere, there's a little intercalated duct. And this is one in cross section right here. You can still see those extremely euchromatic nuclei like the central acinar cells. And then those intercalated ducts will transition into the slightly larger intralobular ducts, like we see one in longitudinal format right here. Okay, so the duct system is intralobular ducts, and there's two portions, the intercalated and then the intralobular. I know that's kind of redundant because intercalated is also intralobular, but that's the way it is. So we've got the little intercalated duct, and then that transitions to the intralobular duct, and then that transitions to the INTER, the between the lobules, interlobular ducts, and then out to the major duct, that would be the pancreatic duct or the accessory pancreatic duct. Then we have the secretory units, the adenomeres that are tubuloacinar, filled with acinar cells that are very basophilic cytoplasm, euchromatic nucleus, have all these digestive enzymes that they're waiting to release. And these are these pre-enzymes or proenzymes that aren't activated until they're released into the lumen of the duodenum, and that pH is raised by the secretions of the intestinal glands. I'm going to stop there. I know you've got the quiz on your mind, but that was an introduction to the pancreas. We'll kind of review it again quickly before we start into the salivary glands, and then go over the liver on our last lab next week before the midterm, which would be the week after. All right, Dr. K.
That sounds good. So what we'll do is we will go ahead and we will go into a breakout rooms.